Welcome to Online Lecture 3, which covers Chapter 3, Nucleic Acids, Proteins, and Enzymes. The pie chart on the right of your screen shows the relative abundance of the major macromolecules in living organisms. You can see that the most abundant of these are the proteins, followed by the nucleic acids. So this lecture focuses on these most abundant macromolecules. And you may recall that we covered the other two groups, the carbohydrates and the lipids, in Chapter 2. Nucleic acids are informational molecules. They're called this because they store and transmit genetic information. In terms of their chemical structure, they are long polymers of nucleotides joined by covalent bonds. Let's look more closely at their chemical structures. There are two types of nucleic acids which differ in structure and function. They are called DNA and RNA. DNA is an acronym that stands for deoxyribonucleic acid, whereas RNA means ribonucleic acid. The monomers that make up nucleic acids are called nucleotides. These consist of a sugar covalently bound to a phosphate group and a nitrogenous base. The sugars found in nucleic acids are 5-carbon sugars, or pentoses. RNA contains ribose, hence the name ribonucleic acid, whereas DNA contains deoxyribose, hence the name deoxyribonucleic acid. The difference between these is that deoxyribose has one less oxygen than ribose, which gives rise to the name deoxyribose. These sugars are bound to nitrogen-rich compounds, or bases. The nitrogenous bases comprise two groups, which each have a distinct chemical structure. The pyrimidines include cytosine, thymine, and uracil, whereas the purines include adenine and guanine. These two groups differ in the number of carbon rings. And note that uracil is found only in RNA, whereas thymine is found only in DNA. And I know that some of you may have trouble keeping the names of those two groups separate, so I'll tell you a little trick that I've used. You'll notice that the purine are the compounds with the double ring structure, and so I always remember that purine, which has 2 syllables, corresponds to the 2-ring structure, whereas the pyrimidines, you'll notice, have only a single carbon ring. I hope that helps you remember. It certainly helped me. In both types of nucleic acids, the polymers are formed by condensation reactions that result in covalent bonds between the sugar of one nucleotide and the phosphate of another. This results in the formation of a backbone of alternating sugar and phosphate groups, called a sugar-phosphate backbone. What's interesting about this is that in every strand of DNA, the sugar-phosphate backbone is identical, and in every RNA strand, that sugar-phosphate backbone is identical to other RNA molecules. So you might wonder, how do these carry information? Well, that information actually comes in the sequence of the nitrogenous bases. There's incredible variation there, and this precise sequence of nitrogenous bases differs between genes, between individuals, and between species. And that's where the true information is. So it's in the sequence of nitrogenous bases, not in the backbone itself. And I'd like to point out something very special about DNA and RNA molecules. And that's that these molecules have what we call directionality. So you can't just put nucleotides together in any order. In fact, if you'll look at this diagram, you'll see that the way that this little stretch of DNA is drawn here has a 5' phosphate at the top and a 3' hydroxyl group at the bottom of the screen. These nucleotide polymers are always formed by a ribose or a deoxyribose being added to this 5' phosphate. And so when we talk about during either DNA replication, which is when DNA strands are made, or in transcription, which is when new RNA strands are made, when we talk about the direction of those reactions, we say that they occur in the 5' to 3' orientation. This is fixed and it never varies. Whenever you see a DNA or RNA sequence written on the page, you should always see someone write the 5' at the beginning and the 3' at the end, but if they leave it off, you should know that that's implied, although we never want to write it that way. We always want to indicate it. So let me show you a simple example here. So here's an example of how DNA should actually be written, and the sequence of the nitrogenous bases are all that's written, because remember, that's where the information is. So this is a short sequence, and the bases occur in the order ATT, GACT, from the 5' to 3' orientation, with those abbreviations being for adenine, thymine, guanine, and cytosine, respectively. If this strand were to grow, the bases would be added on the 3' end, like so. The same convention holds for an RNA sequence, but of course, because there's no thymine in RNA, you will see the U's instead to indicate uracil. Okay, so now I'd like you to take a break and just get out a piece of paper and really quickly just make a list that compares and contrasts the differences and similarities in the structure of DNA and RNA that we've covered so far. So go ahead and hit the pause button. Okay, so hopefully your answer looks something like this. So in terms of the similarities between the two, they're both long polymers of nucleotides, they're informational molecules, and they're both synthesized in the 5' to 3' direction. In terms of their differences, DNA contains deoxyribose and the 4 nitrogenous bases shown here, whereas RNA contains ribose and uracil in place of thymine. If you got those correct, good job. If not, you may want to go back and review the previous slides. RNA and DNA also differ in their overall three-dimensional or macromolecular structures. Not surprisingly, these differences in structure give rise to very different functions. These pictures show the most obvious differences in structure. DNA, shown over here on the left, has a very regular shape, while RNA is quite irregular. The macromolecular structure of DNA most commonly found in cells is called the alpha helix. To understand the alpha helix, think of the shape that would result if you were to twist a ladder. In DNA, this ladder actually consists of 2 strands of DNA that are held together by hydrogen bonds between the nitrogenous bases. When you're looking at a double helix, you're actually looking at 2 different polymers of DNA, each with its own sugar phosphate backbone that's covalently bonded. These 2 individual polymers are held together by nitrogenous bases and the hydrogen bonds between them. In this diagram, the sugar phosphate backbones are indicated by these uncolored structures here. There's one on each side. And the nitrogenous bases are shown by colors in the middle. The number of hydrogen bonds between each set of nitrogenous bases is indicated by the dashed line here. I also want to point out to you that there is directionality to both of these molecules. You can see that on the one on the left, the 5' phosphate group is at the top and the 3' hydroxyl group is at the bottom. And if you look closely at the strand on the right, you'll see it's actually in the reverse direction. And this is indicated by the arrow shown here and also the arrow that's shown over here. Because of that, we call DNA molecules as being oriented in an anti-parallel fashion. The hydrogen bonding occurs only between certain pairs in what we call a complementary fashion. In complementary base pairing, purines always bind to pyrimidines. So for example, if you take a close look at this figure, you'll notice that every one of these base pairs has a double ring structure and a single ring structure that are sharing these hydrogen bonds. So in this case, on the bottom here, this green indicates a purine and the yellow indicates a pyrimidine. This is an adenine-thymine base pair. The other kind of base pairing that we'll see is a cytosine-guanine base pairing, and that's indicated by this red and blue pair. Here's cytosine on the left and guanine on the right. And you can always distinguish these, what we call AT base pairs, from the GC base pairs by the number of hydrogen bonds between them. Every GC pair has 3 hydrogen bonds, whereas every AT pair has only 2 hydrogen bonds between it. One thing that I find very interesting about these hydrogen bonds is how that it is that such a weak bond can hold together 2 DNA molecules and allow them to make up what we call a chromosome. Well, the reason that this works is that even though the hydrogen bonds themselves are very weak, there are a lot of them along the length of a DNA polymer. So to sum up, the structure that we see in DNA really results from this base pairing between 2 strands and the twisting of these strands into a structure that looks like the twisted ladder I mentioned at the beginning. What you're looking at in this slide are actually just 3 different ways of representing the same thing. The diagram over here on the left actually shows you some chemical structures and some bonds. The DNA is untwisted and it's laid out flat. On the right-hand side here we have what's called a ribbon model, and the bases are actually just indicated by what looks like those rungs on the ladder, these horizontal gray bars, whereas the sugar phosphate backbones are indicated by blue and also red. So those look more like the arms on a ladder. And on the bottom of your screen, what you see here is called a space filling model. And this is actually the same kind of shape that you're looking at up here on the upper right-hand side, with the difference being that the sizes of the spheres actually indicate something about the sizes of the various atoms. And so remember that in all DNA molecules, this sugar phosphate backbone is invariant. It's just a repeating groups of sugar and phosphate, with the only difference being their orientations and the 2 different strands that make up a double-stranded DNA molecule. The variability, of course, comes in that sequence of nitrogenous bases, as we've already discussed. So, to sum up about DNA, it is a double-stranded molecule that is held together by hydrogen bonds between complementary base pairs. It contains adenine, thymine, guanine, and cytosine. So now we're going to turn our attention to RNA, which, as I mentioned before, has a quite different macromolecular structure. RNA, as you know, consists of the 4 bases and has uracil in place of thymine. It still has a sugar phosphate backbone, but despite these similarities, RNA has a very different macromolecular structure. The primary reason for this is in the fact that it's single-stranded and that it does not join with another strand, as we saw in DNA. Although interestingly, because RNA is single-stranded, it can actually fold extensively to form intricate structures. This folding is made possible by the same kind of base pairing that we saw in DNA. The difference is that in RNA, the base pairing actually occurs between a single strand, which folds up and pairs with itself. This slide shows the structure of a ribosomal RNA. It's only shown in 2 dimensions. If we were to see the actual 3-dimensional structure, what would happen is that these 2 areas that are shown in blue, the one here and the one over here, would actually fold up together in the direction that's indicated by the arrow. And you can see that this is a very complex structure. It consists of areas where there is base pairing. These are the same kind of complementary base pairing we saw in DNA, with the exception that we see adenine, uracil, or AU base pairing because thymine is absent in RNA. And in the region where that base pairing occurs, those are what are called stems, and in these open regions, those are called loops. And when you get a little loop like this at the end of a stem, the entire structure is referred to as a hairpin. So you can see in this figure that this RNA has a very complex macromolecular structure that consists of stems and loops and 3-dimensional folding. So ultimately, the shape of any individual RNA is a function of the sequence of its bases. This is very different from DNA, which has that regular helical structure. Let's turn our attention now to the unique functions of DNA and RNA. DNA is the hereditary information of the cell. In fact, Sperm consists of little else. It's basically DNA with a tail. DNA contains all the information necessary for growth and reproduction of living organisms. It's copied every time cells replicate, just prior to cell division, and of course that process is called mitosis. And here's an example on the right-hand side of your screen of a cell that is undergoing mitosis, and the genetic material, the DNA, is stained red, and you can see here in a stage called anaphase that the two sets of chromosomes are being pulled apart to opposite ends of the cell. But what's interesting about DNA is that it doesn't actively do the work of cells, and so it's best to think of DNA as a reference book. A lot of information is there, but the book itself doesn't do very much work. In contrast, the cellular work is done by RNAs and a macromolecule called a protein, which we're going to discuss in the next part of online lecture 3. What's interesting about them is that the information to make them is actually held in the DNA. So RNA actually arises from a process called transcription. This is indicated on the left-hand side of your screen here. So here's our double-stranded DNA. This is our library of genetic information. And through a process called transcription, a piece of RNA is made from the DNA instructions. This process of transcription relies upon complementary base pairing. So a DNA original or template is actually read by an enzyme which just incorporates the matching base into a piece of RNA. Once the RNAs are transcribed, they can have a variety of functions. For example, that ribosomal RNA that we looked at earlier, when it folds up, it actually joins with a bunch of other RNAs to help form the structure of a ribosome. This slide is showing you the two subunits of a ribosome, and all these different colored structures you see, the grays, the purples, the yellows, and the oranges. Each of these are a separate RNA strand that has folded up and joined with others to provide a structure. In the ribosome, the RNAs also have a special kind of function. They actually serve as a type of enzyme called a ribozyme and have catalytic activity. So the RNA in a ribosome has both a functional and a structural role. RNAs can also specify the sequence of amino acids and proteins. So they form the template that's used in a process called translation, where a ribosome actually reads a special kind of RNA called a messenger RNA and uses the information there to make a sequence of amino acids that ultimately comprise a protein. So just in the picture that you're looking at here, there are three different kinds of RNAs. There are special RNAs called transfer RNAs, which help bring in the amino acids that make up the proteins. Here's a messenger RNA, which has the instructions that specifies the sequence of amino acids that should occur in that particular protein. And also inside the ribosome, there are those ribosomal RNAs that we just looked at in the previous slide that are actually forming that structure. So RNAs have quite a bit of diversity of structure and function. And in addition to the ones that are shown here, there are other RNAs that can have a regulatory role, such as short interfering RNAs or microRNAs that are involved in controlling the expression of genes, because not all genes are turned on all the time. That would be terribly inefficient for living organisms. We'll cover that information in later chapters. That concludes part 1 of online lecture 3. Be sure to watch the other parts as well.